Welcome, everybody. My name is Richard Miller. I'm the CEO of Corvus Pharmaceuticals, and we're delighted to be conducting this webinar and in-person meeting at the Society of Investigative Dermatology meeting, where Corvus and its collaborators have two papers, two posters, and the pleasure of having two of our presenters speak here today with you. So this is a very exciting time for Corvus. I think you'll see both from some of my introductory remarks and from the speakers that we have here today that Corvus really does have a very special compound that's going to have applications in many, many areas of medicine. And so let me start with our forward-looking statements. And introduce today's speakers in addition to myself. I'm very happy to have Dr. Albert Chu here. He's a clinical professor of dermatology and the director of clinical research in the Department of Dermatology at Stanford University Medical Center. Along with Albert, Dr. Kavita Saran. Kavita is a professor of dermatology and an expert in precision medicine and genetics, also in the Department of Dermatology and also in the Cancer Institute at Stanford. Now, I picked these two speakers for a reason. Yes, they're very smart. Yes, they're very photogenic. But more importantly, they complement each other. That is, clinical research together with the science and elucidation of mechanisms, which is going to hold for us the promise of new applications and better ways to use our drugs and understand them better. So these two speakers really complement each other and really add to the scientific prowess of Corvus. They make us smarter, and they set new standards, higher standards for us every day. Let me start with the agenda for today. I will make some introductory remarks, and then we'll have Dr. Albert Chu talk about the treatment landscape for atopic dermatitis, as well as review the clinical data from our phase 1 trial. Now, interestingly, we heard this morning from Kavita on the biomarkers. That was probably would be better to have the clinical first, but the schedule of the meeting, we can't control. So Dr. Saran went first, but she gave a brilliant talk and is going to go through that again for us. So after Dr. Chu, Dr. Saran will go through the immunologic and biomarker data. And I use the word immunologic deliberately. It's more than a biomarker. It's fundamental biology. We have learned new things about this disease and about the immune system. Then I'm going to follow Dr. Saran's talk with some key takeaways, if I'm able to summarize their beautiful work, as well as talk about our pipeline. And then I hope we have time for Q&A, both from the people in the room and those in the back. You guys can move up here. We have more seats up in the front. So we'll have Q&A from people in the room here, as well as on the webcast. And I know we have a very large audience on the webcast. Okay, so with that, just some details on the papers that we're talking about. On the left, Dr. Saran's talk was this morning, and really beautiful talk. I hope that you can look at her slides and go through it, but she is going to cover most of that today. There's a poster session this afternoon with her data, and she'll be there and able to answer questions in more details if you have any. And then on Saturday morning, Dr. Chu is going to deliver his paper on the phase 1 clinical results. I think that's going to be a very important paper. The poster on that paper will be tomorrow afternoon as well. So a lot of details that they're not able to cover in the usual 10-minute talk. Okay, so let me start with sort of get everyone grounded. Soquelitinib is a first-in-class drug. Why is it a first-in-class? Because other people have talked about ITK, a target, and I'll get into that more in a moment. But really, nobody was able to make a drug that just hits ITK. And that's because it's hard to do. ITK has several closely related enzymes called the Tec family kinases. And some of those just have one or two amino acid differences in their structure and their ATP binding site. And I'll tell you in a bit why we were absolutely fastidious about the specificity. So we have a novel mechanism of action. First-in-class drug with, we believe now, broad opportunities because it can control so many important functions in the key immune cell called the T cell. So the mechanism of action is dependent on a high degree of selectivity for ITK. And this enables it to block many different pathways, also to reprogram T cells so that you can have a lasting effect beyond the drug. Kind of like when you vaccinate somebody. They're vaccinated, the drug is gone, but you're immune for a while. Well, this is sort of the same thing. The immune system has memory. It's programmable. Thank God for that, because otherwise we'd have to take medicine every day. Oral administration. We're studying this drug mainly BID dosing. We believe eventually it's a QD administration. It's now in a phase 3 trial in a very bad disease called peripheral T-cell lymphoma. It's a registration randomized trial in peripheral T-cell lymphoma. As well, we're now enrolling our phase 2 trial, randomized double-blind study, phase 2 in atopic dermatitis, similar to the patients that you're going to hear about in a few minutes from Dr. Chu. We plan to start studies in other diseases, hidradenitis suppurativa later this year and asthma. And that's just the beginning because we think that this really is a pipeline in a product, and I know that's a cliche, but this really could be that. I had the privilege of working on an antibody called Rituxan. That was a pipeline, a product in a pipeline too, because it could do all different kinds of lymphomas. You could treat various autoimmune diseases. So I look at this in a similar way. The opportunity is vast. Pulmonology, gastroenterology, dermatology, oncology, rheumatology, just to name a few. Strong IP. We have issued patents, composition of matter patents. It's issued in all the major territories. You can look up the patents. Those protect us in those territories through to 2042. And we have a bunch of patent applications covering things like methods of use, monitoring, TH2, TH1, Tregs, etc., all kinds of other permutations of that, working our way through the USPTO. Management team. So we've done this before. My colleagues and I have done this before. It's called Rituxan, it's called ibrutinib, and a couple of others. BRAF inhibitors. So we know how to take drugs or biologics that have big opportunities and get it done. And we're all driven by science, medicine, good clinical research, and development. That's what we pride ourselves on. Okay, now let's talk a little bit about the mechanism, because I think we can make some really important points here. So ITK stands for interleukin-2 inducible T-cell kinase. It's been around a long time, as I mentioned, and it's known to be crucial in many T-cell functions. T-cell receptor signaling, T-cell differentiation, apoptosis, many T-cell migration. It's important in many areas. And we know that it's fundamental in the differentiation of a naive CD4 cell into what are known as Th17 cells, Th2 cells, and Th1 cells. So those are the three main types of helper T cells. The Th1 cell, that's an important cell, fights cancer, fights infection. Th2 cell, important in protecting also against certain infections and tissue homeostasis, regeneration, same thing for Th17. Now Th17 and Th2 cells are the cells we're going to focus a lot on today because they're involved, they're heavily involved in inflammatory diseases. So it was shown by Pam Schwartzberg and Leslie Berg 25, 30 years ago that if you knock out ITK gene in a mouse, just ITK, what happens, as shown on the right here, is that you can't make Th17, you can't make Th2, because they're dependent on ITK. And then, consequently, you can't make those cytokines. What are those cytokines? IL-4, 5, 9, 13, 31, GM-CSF, TNF-alpha. When you think neutrophils in some of these diseases coming from there, GM-CSF. But, interestingly, when those workers made that knockout mouse, they still were able to produce Th1. Why was that? Well, the Th1 cell has a redundant enzyme known as RLK. Sometimes, in the old literature, it's TXK. You may see that terminology sometimes. Okay, so the genetic studies showed that if you knock out ITK, you skew to Th1, you lose Th2, Th17. Now, interestingly, I know there's a lot of companies out there that are saying, I got ITK inhibitor too. Most of them hit RLK. The genetic studies show if you hit RLK and ITK together, you get T-cell dysfunction. You're severely immunocompromised. And of course, we don't want to do that in autoimmunity. So, what we did at Corvus is say, we want to do this. So our strategy, the inventive step, the aha moment, every great company has an aha moment, was, hey, we got to make it specific for ITK and not for RLK. And we weren't really that smart, even though we were saying aha. We were not that smart. We just went to the library and read a lot. That's helpful. So anyway, now, the important thing to tell you about interleukin 2 inducible T-cell kinase is that it's not all over the body. It's just in T cells and NK cells and ILC2s. We don't have time to talk about ILC2s today, but they're really important in immunology and in autoimmune diseases, asthma, atopic dermatitis, other diseases. But the specificity of the drug for a target that is only in T cells and NK cells. So that's really, really very important. So when we hear about mechanism, you're going to hear a lot of stuff about mechanism from Dr. Chu and Dr. Sarin. Remember, it's limited to those tissues, to T cells, and T cells can orchestrate all these different kinds of immune effects. I told you memory was part of the immune system. There's a second important component, diversity. It can do a lot of things. Because one day you're getting a virus, the next day you're getting a bacteria, the next day you're getting a fungus. You don't know we're getting assaulted every minute. Trust me on that. So, the strategy, make an ITK specific drug. And we were able to do that. Okay, and we've published that. Alright, we have an oral ITK specific drug. It is a covalent drug, by the way. That's one of the ways we confer specificity. I wouldn't go into the details of that, but this morning there was a question about covalency. Well, yes, it's covalent, but the protein turns over in 12 hours. So the drug is out of your body very quickly. The half-life of this drug in the body is, in the plasma rather, is 2-3 hours. Okay, so there's no lasting effect of the drug once you stop it. Pretty much gone. In a mouse where we can give radio-labeled compound, we know 99.99% is gone in 24 hours. Anyway, so this is the mechanism of action. So if we can make a drug that can block ITK and spare RLK, we may inhibit Th2 and Th17 and spare Th1. That's a good thing. Okay. Now, each of these cells has characteristic transcription factors. So we can elucidate them. We can measure them. We can look at them in the body, in the blood, and so forth. GATA3, ROR gamma T, T-bet. You'll hear about another one pretty soon called FOXP3. In fact, you're going to hear about it right now. So the story got much, much stronger in the last few years. Now there is no question No question whatsoever. There was just another paper last week that came out from a group in New York that Th17 and Tregs are suppressor T cells. They're the brakes on the immune response. Anytime you react to a microbe or whatever, you make an inflammatory response, but sometimes you've got to shut it down when the microbe goes away. Those are the Tregs, among other things. So ITK turns out to control the differentiation of Th17 and Tregs. And when you block ITK, and this was done by Avery August using our drug, it was also done by him genetically, you stop Th17 and you skew towards the Treg. There's no question about that anymore. We've seen that in every experiment we do in the lab. 5 or 6 different labs have shown that. And we now see it in our patients in atopic dermatitis. Oops. Okay, so where does this leave us? Well, we have a drug, very specific, for a target that's only in your T cells, which is not all over the place. It's very restricted. And yet we have the ability to block various cytokines, the Th2 cytokines, 4, 5, 13, etc. Th17 cytokines, like IL-17 and 21 and GM-CSF and others. And we can increase Tregs. I'm not going to go into the ILC2 story here, but that's an emerging, very important part of the story. Now, comparing that to some of the approved agents, which you already know, those are stoichiometric. If I give an anti-IL-13, I'll block IL-13 for sure, but I won't block the other things. And I'm not really changing the function of the immune system. I'm blocking a ligand. Now, JAK inhibitors, of course, they can modify T cell responses, but many of these targets of the current drugs suffer from the fact that many tissues express the target, notably the hematopoietic system and the immune system. So if we look at some of the stats or some of these other receptors or proteins, signaling proteins, they're not just in T cells. They're in T cells, B cells, myeloid cells, erythroid cells, et cetera, et cetera, et cetera. I can't name them all. I don't remember them all. But they're throughout the system. And that gets a little scary if you want to treat people for long term, if you wanted to do that with agents that are perturbing fundamental processes in the body. Okay, so that's the foundation here. The foundation for taking soquelitinib into the clinic in immune diseases. We were already in the clinic in T-cell lymphoma. Now, the reason why T-cell lymphoma is attractive is because those are Th2 cells. They frequently involve the skin, and they're really going crazy. They have no respect for the patient. They grow and grow and grow. We think we can inhibit them in a large number of patients, and that's a phase 3 trial that's going on now. But along the way, as we began to put pieces of the puzzle together, and as we started to do more experiments in the lab and preclinical models in animals and so forth, we began to realize that autoimmunity and inflammatory diseases would be great. And that led us to atopic dermatitis. Th2 disease involves the skin, many of the same cytokines involved, and you can see the disease very easily. Plus, I was friendly with Albert and Kavita. No, not really. I'm sorry, I am friendly with them, but that wasn't the reason. Anyway, so with that, is my great pleasure now to turn the podium over to Dr. Albert Chu. And I think you're going to find his talk extremely exciting.
Just a quick mic check. Is this coming through? Okay. Great. My pleasure to speak here, and I actually really love this format because I get a little more time than at the actual symposium talk to double click on the details here. So like Richard mentioned, my name's Albert Chu. I'm a medical dermatologist over at Stanford. I serve as our department's director for clinical research, and I've been involved in clinical trials for atopic dermatitis including some of the early dupilumab trials, the IL-13 inhibitors, a number of the JAK inhibitor trials, and then now Soquelitinib. So I also take care of a large number of patients with moderate to severe atopic dermatitis who are on systemics, including a busy practice where we see a lot of the folks referred in from the community who have failed first and second line therapies. So this is a topic near and dear to my heart. And maybe I'll just start first with a reflection. Atopic dermatitis it's not a lethal disease, but it's a highly, highly morbid disease. It is really tough on our patients. So this is a combination of factors. One is that, especially in our adult patients with moderate to severe disease, for most of them, this is going to be a chronic, lifelong issue. They deal with very unpredictable flares, and that in itself is very, very hard on people psychologically. And then you just have this daily bother from itch and frequent complications like skin infections. So it really hits a lot of aspects of patients' quality of life, their health. And what's really interesting about this patient population is there's a lot of varied patient preference on what is the ideal treatment for them. Even in my most severe patients, what I find is that we're oftentimes balancing these discussions of efficacy, but also what each individual is willing to accept in terms of side effects. There's also a lot of variation in what they'd accept in terms of treatment duration and what happens if they miss a dose or come off the treatment. So with this context, I'd love to talk through the therapeutic landscape right now, at least our approved therapies that we're using right now in the clinic. So first and foremost, we have to acknowledge dupilumab, our first real targeted treatment, and that in our IL-13 is now the mainstay first line for atopic dermatitis, and that's primarily because of the excellent safety profile. They don't have broad immunosuppressive effects. So in this disease, safety is paramount. And one thing we have to keep in mind, I'm an adult dermatologist, and I know a lot of our need is in adults, but a lot of this disease burden is in children, where safety is even a higher bar. That being said, with these biologics, we know the targeted monoclonal antibodies, there can be variable response. Remember, depending on which of the dupilumab trials you take a look at, about 30 to 60% of patients don't hit EASI-75, which is a high threshold of clearance of the skin. Also, patients have to be comfortable self-injecting themselves with subcutaneous medications, and that just adds to patient hesitancy, requires a lot of hand-holding from our staff. And then finally, they do have side effects. So if you look at dupilumab, the IL-13 inhibitors in particular, conjunctivitis, injection site reactions, these are all things that come up when you talk to patients about this. Now, for the patients who want an oral medication or essentially are reaching for a little bit more efficacy, the JAK inhibitors have become really, really helpful tools in our toolkit. That being said, we have to acknowledge their limitations. They have a really hefty black box warning that infections, have to talk about infections, blood clots, death. You can imagine this really adds to the hesitancy that patients have about starting them. And then they require regular blood monitoring, which again, just another barrier to patients willing to start this. And so I do think that with all this here, there is a tremendous need for additional treatments for atopic dermatitis, especially those that can address some of these aspects of the disease I just mentioned. So first and foremost, we have to acknowledge that there are a lot of patients who don't respond to any of these first or second line therapies or have some side effect or contraindication to them. So we always need something with a novel mechanism of action that can capture some of these patients and rescue them. So that's just very, very clinically important. But you can take this a step further. Let's say that, imagine that you're a fly in the wall for one of these conversations with these patients where they're first indicating their interest in moving beyond topical therapies, trying to get into a systemic therapy. And I think the most frequent refrains you'll hear are something along the lines of, hey, is there a safe pill that I can try for a while? Or very rational questions like, if I start this medicine, am I stuck on this for the rest of my life? Or what happens when I stop it, when I decide that I don't want to take it anymore? And so we're, in this current landscape, having a lot of these nuanced conversations about trade-offs. And we are talking about potential side effects, blood monitoring, if you're talking about the JAK inhibitors, if you're being really honest, you have to say, gosh, we have great data that if you stop this medicine, you have an 80% chance of flaring and needing some sort of rescue medicine, potentially within weeks. So that's where we're at. And I think if we take a step back, we are in a far better place now for atopic dermatitis. And when I first started, when we're just using conventional immunosuppression. But that being said, I think our patients are asking us to do better. And that's why I think it's so important that we have clinical trials like this and in development of treatments like soquelitinib that can offer more to this really large and important patient population. So with that, I'll jump into the study design and clinical data that we're presenting here at the SID. So here's the study design. This is a randomized, placebo-controlled, blinded dose exploration study looking at soquelitinib for moderate to severe atopic dermatitis. You can see here that we have 72 participants, and kind of the standard entry criteria for moderate to severe atopic dermatitis that is focused on adults here. Patients who are required to have failed a prior topical or systemic therapy, and that will come into play later when we talk about the prior systemic exposures. So for the first two cohorts, cohorts 1 and 2, patients were evaluated for soquelitinib at the same total daily dose, but either at split or once daily dosing. And then cohort 4 essentially doubled that dose to evaluate them at the highest tested dose in this trial. Cohorts 1 through 3 were randomized 12 patients to active treatment and 4 patients to control, and they were treated with just 4 weeks of active treatment. Cohort 4 here, I'll kind of point out, is an interesting one. There are a total of 24 patients in this cohort. They were randomized 1-to-1 active and placebo. And then these patients were actually evaluated for 8 weeks of total duration of treatment, so giving us a peek at what happens with an extended course of treatment. So here are our patient characteristics here at baseline. And you'll see here, these are cohorts 1 and 2, cohort 3, a pooled cohort 1 to 3, and then the cohort 4, which again, those are the patients that received 8 weeks of total treatment, so twice as long as the other 3 cohorts. Kind of if you just look closely at the actively treated patients versus the placebo-treated patients, you'll see they're actually well balanced between them in terms of baseline demographics, baseline EASI scores, and proportion of prior systemic therapy exposure. One of the things we did observe is that as we got into cohorts 3 and 4, the baseline EASI severity actually crept up, and we think that's basically due to the nature of the recruiting sites that activated by that point in the trial. I think one interesting point about this trial population is a fairly high proportion of patients who are exposed to systemic therapy, so more than a third, which is not the case in many of these atopic dermatitis trials. When most of those prior exposures were dupilumab, but for representation of other AD systemic therapies. So let's jump into the efficacy results. This is seen here for cohorts 1 through 3. So recall these are the patients treated for 4 weeks of total dosing. And we'll take a look here for cohorts 1 through 2, looking at EASI-75 and IGA 0/1. So these are really important endpoints. They traditionally have been co-primary endpoints for pivotal trials for atopic dermatitis. And we can see for the lowest tested total daily dose, cohorts 1 and 2, we're actually starting to see patients hit these responder thresholds at just 4 weeks. And then if you take a look at cohort 3, these are the patients who received double the total daily dose compared to cohort 1 and 2. You'll see an even more impressive EASI-75 and IGA 0/1 responder rate there, particularly if you look at EASI-75. And notably, for these patients, none of the placebos were able to hit any of these responder thresholds. If you take a look at EASI-50, EASI-90, and the mean percentage reduction EASI, you'll see very similar directional results with improving scores for cohort 3 at the highest dose, especially when you compare it to placebo. So here's a plot of the mean percentage reduction in EASI for cohorts 1 through 3. And what you'll see here on the vertical axis is the actual change in EASI percent from baseline. And then horizontal axis is time, of course. And then I'll just point you towards that middle white period. This is the actual 4 weeks of active treatment that these cohorts 1 through 3 patients received. And all the way on the right in the gray here is a 30-day follow-up period off any active treatment. So they're followed up 30 days after the last dose to see how their skin and safety is doing. So in orange here is the placebo group. This is a pooled placebo group from cohorts 1 through 3, and you'll see them in the top curve there. And then in blue and red here are cohorts 1 and 2. Again, these are the patients that got treated with the same total daily dose. And you can see that their curves overlap almost perfectly, and they do start to separate from placebo. You can start to see that occurring at day 15, and that becomes very apparent on day 28. So here's the really interesting part about this trial. At the follow-up, you actually see maintenance of that clinical response. So I was involved in the other trials for JAK inhibitors and other therapies. This is something that you don't often see in atopic dermatitis, where the disease frequently comes back after treatment. And then in cohort 3 here, in the green, what you can start to see is a dose response. So these are the patients treated with double the total daily dose. You have a earlier and deeper response. You can see it as early as day 8, statistically significant day 28, and again, that maintenance of response here. So we'd love to double click on that part of it, because as a clinician, that's what really popped out to me with those data. First of all, the efficacy, of course, the rapid deep response, but also this durability. So what's interesting in this slide is that this is a real focus on cohort 3, and these patients, unique to this cohort, were followed up to 90 days after the last dose. And so what we can see in this cohort, you have that early and deep response, but then you actually have durable maintenance of that improvement all the way to the last date of follow-up. So we actually don't see deterioration even through the last follow-up visit. So we don't know how long this persists because we've ended it and it's continuing. So I don't want to steal Dr. Srin's thunder, because she's going to help us look into the biomarker and potential mechanistic explanations for this durability. But I will just point out too, if you really look at this cohort 3 population, you'll actually see that they have increased T regulatory cells in the blood, and that's especially apparent when you compare them to placebo and the lower dose treated patients. So if you recall from the mechanism, we know that ITK is activity shifting us from Th17 inflammation over to those T regulatory cells. So that could provide a potential mechanism for why we see this clinical durability. So this is just a reminder of what happens in real life when you treat patients with other medications with this rebound or worsening of the skin disease. So these are 3 of our approved therapies, and just an example of an investigational product. So the JAK inhibitor is kind of a classic example, highly efficacious, but in the clinical trials and just clinical experience, you can see once you stop the medication, that skin improvement starts to deteriorate pretty rapidly, oftentimes within 2 to 4 weeks. And then just anecdotally, my patients tell me that they miss a dose, they'll feel that itch starting to come on even before the signs start to change on their skin. Even with dupilumab, which is a biologic, we know from the solo continuation studies and just from clinical experience, this is where day 0 is actually when they completed their 16 weeks of dupilumab and they were randomized to other doses of dupilumab, or essentially stopping it by being switched to placebo. And you can see that deterioration in the skin control at that point as well, seeing hints of that as early as 4 weeks, but definitely by 8 weeks. So this is just one more example of another investigational product, the STAT6 inhibitor. And what you can see there is that at the end of treatment, you can start to see some of that clinical deterioration as well. So now let's take a look at cohort 4. And I actually really love this slide because this gives us a sneak peek at what happens if you try to extend the duration of therapy. So cohort 4 is unique in this study. These are the patients that got 8 weeks of active treatment. Again, a 1-to-1 randomization, active to placebo. And then here we see the highest degree of EASI-75 and IGA 0 and 1 response. At this 8-week treatment duration, we are seeing some patients in placebo hitting EASI-75, but very, very low compared to the actively treated patients. And we see directional results in the same manner, EASI-50, EASI-90, and a mean reduction in EASI. But again, just really pointing out 75% of these patients are hitting EASI-75, which is, as a clinician, something you're really excited about. At this treatment duration as well, we're starting to see the placebo patients flare as defined by requiring rescue medications. So we had a couple patients that flared here and none on the soquelitinib treated. So just plotting out here the response curves, and what you can see here in orange is the placebo group, and then what we see is this continuing to deepen response curve for the cohort 4, again, highest treated dose, also the longest period of treatment, and then that clinical durability seen again at the end of that treatment period. And of note, I will point out that that slope does not start to plateau. So at 2 months of treatment, we do not see a plateau in the improvement of the EASI scores there. So this slide, I think, is very illustrative if you're just trying to take a look at all the different cohorts and kind of their EASI-75 and IGA-01 response. So on the left here, we're taking a look at cohorts 1 through 3, again, treated for 4 weeks. And in the solid bars here, we can see the EASI-75 responder rate. And then in the striped bars, we can see the IGA-01 responder rate. So you can see in this pooled cohort 1 to 3, at the end of just 4 weeks of treatment, we are seeing EASI-75 and IGA-01 response. And then interestingly, that is maintained day 30, day 60, and day 90. And again, we are not just cherry picking the responders here. This is the total population in maintenance over that time frame. And then cohort 4, what you're seeing here is, again, longer duration of treatment. You have a superior EASI-75 and IGA-01, and then maintained at the 30 days of follow-up. So we then ask the question, so what happens to the folks who've had prior systemic therapy exposure? Again, a kind of a marker for more severe disease or tougher phenotypes. And this pools all 4 cohorts together, so you can see the placebo group versus the soquelitinib treated groups, and you see separation of the curves in terms of mean % reduction in EASI. And then over here on the right panel, you can see the patients with prior systemic exposure. So even that placebo group is only the placebo-assigned patients who had prior systemics and actively treated patients with prior systemic exposure. And you can see it continues to have that separation in the curve, suggesting good activity within these patients with prior exposure. And this is the one I love as a clinician. So this is the folks who knock on my door. These are the patients with prior systemic failures. So taking a look again at the patients with the highest total daily doses, they were able to identify 6 patients with prior systemic failure. All of these patients have failed dupilumab, many other additional agents, including some with a couple failed agents. And what they were able to see is 3 of those 4 patients actually improved on soquelitinib, 2 markedly so, and both patients who were assigned to placebo ended up flaring, again, requiring rescue medications. So I think from a clinical perspective, this really gives us a lot of excitement about the potential activity of soquelitinib in patients with prior treatment failures. So just turning over to a summary of our safety here. This is taking a look at cohorts 1 through 3, again, 4 weeks of active treatment, and then cohort 4, which is 8 weeks of active treatment. And what we can see is that the frequency of adverse events is well balanced between active and placebo-treated patients. Actually, in the folks with the longest exposure, placebo ended up with more adverse events. There were no severe or serious adverse events. No patients discontinued due to AEs. So here's the granular safety table. And again, taking a look at cohorts 1 through 3, and then cohort 4 with the longest exposure. And kind of the big takeaway here, again, a mix between placebo and actively treated patients. Again, pretty well spread across. Most notably, there was no hematologic or laboratory abnormalities, and there was no infectious signal in this safety data. Some patients treated with soquelitinib reported a headache. Apparently, that all occurred at a single site, and most of them were transient, lasting less than a day, so hard to read into that. But overall takeaway, a really clean safety profile, especially from our interests with relation to infections and lab abnormalities. So in conclusion, we present our phase 1 clinical data about soquelitinib. We're really excited about this ability to target Th2, Th17 inflammation, and also potentially start to shift into these T regulatory cells for atopic dermatitis. So far, we see a clean safety profile, no infection signal, no lab abnormalities. And again, we can attribute that to the tissue selectivity of this medication. We saw a early and deep response with a relatively short dosing period of just 4 to 8 weeks. Again, this is a phase 1 trial. And again, that speaks to the broad targeting of Th2. And we also see durable treatment effects with patients with prior systemic exposure, including prior systemic failures. And so finally, just I think we'll tee up my colleague, Dr. Sarin here. We're really excited about this mechanism. We think that this might actually provide promise for other conditions with autoimmune inflammatory disease, especially with this potential mechanism of rebalancing immunity. So thanks so much.
So given the time, I think we'll hold questions until the end. It's now my great pleasure to introduce Dr. Sarin, but before I do, I always like to say that, I have something else to add. Before I do, I always say it starts with the patient. What do we observe in the patient? We observe clearing of skin lesions. We observe safety. We observe a durable effect. And then we go into the laboratory and we say, okay, well, how do we explain this? And one of the things I think you're going to learn from Dr. Sarin is the translation here is absolutely beautiful. And portends for, I believe, success not only in atopic dermatitis, but many other diseases as well. So I'm really looking forward to your talks, Kavita. And I also understand that following Albert is a difficult process.
Thank you very much. Thank you, Richard. Thank you, Albert. That was a great talk and really excited about the clinical efficacy. But I get to present the really fun stuff, which is what is happening underneath the skin that explains this clinical response. So it gives me great pleasure to present the immunologic data from Soquelitinib for atopic dermatitis. And Richard did such a beautiful job going over the mechanism, but I do want to just highlight it one more time so that this is the basis of all of the studies that we'll be presenting so that everyone is on the same page. So as Richard mentioned, ITK is a T-cell signaling kinase that's primarily expressed in T cells, NK cells, and ILC2 cells. And ITK plays a critical role in mediating TCR differentiation and TCR signaling. But what's really important to understand is that ITK is not equally required across T-cell programs. Genetic studies have shown that loss of ITK preferentially suppresses Th2 and Th17 responses, but spares immunoprotective Th1 responses. And that is due to compensation from a related kinase called RLK. The second important thing to know is that ITK plays a critical role in immune switching between Th17 and T regulatory cells. Loss of ITK has been shown to shift cells away from Th17 programs into Tregs. And as Richard mentioned, that is the beautiful design of Soquelitinib, which was designed to suppress selectively ITK, but spare RLK, thereby suppressing Th2 and Th17 immunity, but sparing Th1 so that we retain protection against cancer and other infections. And this biology is central to atopic dermatitis, which is primarily driven by type 2 inflammation. And as Albert mentioned in his beautiful introduction to the landscape of therapies, many of the therapies that are currently very effective for atopic dermatitis suppress these Th2 cytokines in this pathway, IL-4, IL-13, and so on. And they are effective, but they're not necessarily durable, as Dr. Chu mentioned. ITK inhibition is happening just upstream at the T-cell programming to shift away from Th2 and Th17 programs, and so really can lead to more of an immune reprogramming or a shift to suppress these downstream cytokines. So Dr. Chu also did a great job going over this. I'm going to be extremely brief and summarize the whole trial in one single slide. So this was, phase 1 placebo-controlled study, and patients were enrolled in 4 cohorts. In the first cohort, patients were treated for a total of 4 weeks at increasing doses of Soquelitinib. And in the fourth cohort, patients were treated for a total of 8 weeks at high-dose Soquelitinib, 200 milligrams twice a day. Now, for this talk, I want to draw your attention to 2 things. The first is that it was placebo-controlled, but the second is that every cohort had 30 to 90 days of follow-up off of treatment, which allows us to look at the durability of response. And I'm also going to summarize all of the work that Albert just presented into a single slide, which really highlights 2 things: the clinical efficacy, which is significant improvement in EASI scores in 4 to 8 weeks of treatment, And the second thing, which is the durability. If you look both 30 to 90 days off of treatment, none of the patients had a recurrence of their atopic dermatitis. No one had a rebound, and no one required any rescue medications. And this is why it's so interesting to peek underneath the skin and see what is happening immunologically. So from an immunologic standpoint, we investigated the effects of ITK inhibition on Th1, Th2, Th17, and Tregs. And the assays were looking at serum cytokines, blood flow cytometry, and PBMC single-cell RNA sequencing. And this is the first finding. Soquelitinib reduces Th2 biology. On the bottom left is PBMC data showing a dose-dependent reduction in proliferating Th2 cells. The Th2 cells are down. On the right, we see a significant reduction in Th2-related cytokines, IL-4, IL-5, and IL-13. We also see a reduction in TARC, a Th2 chemokine, which is directly associated with the activity of atopic dermatitis. But I want to call your attention to the gray shaded area here to highlight a very interesting finding, which is that even 30 days after treatment, so during the drug-free period, the cytokines continue to decrease. The second interesting finding is that soquelitinib led to an increase in persistent Tregs. On the top here, you can see that soquelitinib led to an increase in Tregs marked by CD4 positivity, CD25 high, and FOXP3 as compared to placebo in the high-dose groups, the 200 milligram twice a day, as well as the cohort 4. But here's what's really interesting. Those Tregs continue to persist after the 30-day drug-free period. And this may actually explain the durability of response that's seen clinically. And when we look more closely at the Th17 Treg axis, we see the following. Soquelitinib increased batch 2, which is a transcription factor that's involved in promoting T-cell stability and Treg stability and longevity in the high-dose group. So on the bottom left here, you can see increased batch 2 expression in the high-dose group treated with soquelitinib as compared to placebo. And conversely, treatment with soquelitinib led to a decrease in WR gamma T, a master transcription factor for Th17 cells. So this suggests that soquelitinib may be playing a direct role in regulating that Th17 Treg axis. So what is one potential mechanism behind this? Well, one is that ITK has been shown to sequester batch 2 in the cytosol through downstream phosphorylation. And inhibition of ITK by soquelitinib may release batch 2 so it can translocate into the nucleus and induce transcription of genes such as FOXP3, promoting Treg longevity. The third finding, which is relatively unexpected, was that soquelitinib led to inhibition of the JAK-STAT pathway. On the top, we observed that SOX3 expression increased in circulating T cells, including Th2, Th1, and TC17 cells. On the bottom right, we observed that JAK1 signaling was suppressed, along with STAT6. And this is important because, as Albert mentioned, the JAK-STAT pathway is central to atopic dermatitis, and STAT6 is central to IL-4 and IL-13 signaling. And this was somewhat unexpected because ITK is not known to play a direct role in the JAK-STAT pathway. So we believe this may be happening through sort of the T-cell immune reprogramming away from Th2 and Th17 programs. And this is also important because that suggests that maybe this JAK-STAT inhibition may be more selective in the T-cell programs. So in conclusion, soquelitinib is an oral, selective, covalent inhibitor of ITK that spares the related kinase, and it appears to be rebalancing Th2 and Th17 responses, suppressing Th2, and actually shifting Th17 responses towards a Treg program with persistent Tregs even after the drug is discontinued. It also appears to inhibit the JAK-STAT pathway in the T cells. And together, this may explain the durable remission that's seen clinically, as well as its favorable safety profile. But more broadly, this really highlights a new paradigm for treating inflammatory conditions. Target upstream at the T cell differentiation node, shift away from the inflammatory T cell programs while preserving the immunoprotective T cell programs, reprogram immunity, and the potential for durable control. And with that, I'd like to turn it back to Richard.
As I told you, seldom in my experience in medicine do you see such a consistent translation where all the things just sort of line up with the proposed mechanism of action and, more importantly, the clinical findings. So that's really beautiful. So I have the task, and I want to go quickly because I do want to leave time for questions. So why are we excited about soquelitinib? It's very simple. We may be modifying the underlying biology of the disease. Okay, we're not just blocking a cytokine or blocking a receptor. We're changing the disease. We're disease modifying. That is a big, big statement, and if is confirmed in other trials and other diseases, really does change the paradigm, as Dr. Saren's last slide mentioned. Now, it's even better than that, because do you realize that those Tregs are antigen specific? Okay. They're not just suppressive. A Treg has a T cell receptor and is only suppressing a particular antigen response. Did you realize that the Th2 cells that were decreased were, as she said very cleverly, proliferating Th2? Because we can figure that out with our science. So it's not just blocking every Th2 in your body, it's the proliferating ones. Those are presumably the ones that are associated with the ongoing inflammatory reaction. Okay, so the precision, and she's an expert in precision medicine, is really, really amazing. So we may be modifying the underlying biology, and that represents a new way to think about therapeutics, treatments, monitoring, etc. And is it worth it? You bet it is, because I don't know of any patient who wants to take a drug forever. Every drug has side effects. Every drug has chronic implications that may take years to figure out. Most people want to take a drug for a while and go off of it, hopefully never come back. If it does come back, do it again. My own personal experience as a physician at Stanford treating patients with Rituxan was patients got 4 weeks of Rituxan, then it went to 8 weeks. Then 70–80% didn't need it again for 30 months. But when they came back, guess what? They wanted it again. And they wanted it again after that. And I had patients in my clinic who I gave Rituxan to 10 times. Because it worked, it was well tolerated, and it gave them a good remission. They didn't have to take pills and come in and do all kinds of stuff. They had a simple infusion, they were done. So new paradigm, we're modifying the disease. And I don't need to go through the details here. You've already heard blocks Th2, Th17, rebalances Tregs. I do want to emphasize that those Tregs are not just suppressive. They're antigen-specific suppression. That's why it was the Nobel Prize last year in medicine. Not for me, unfortunately, but for the people who discovered it. The profile for us now that's emerging, I want you to think about this, is a shorter treatment period. 28 days gave you a durable response. Maybe a longer duration will give you even more durable response. That's a question that will need to be answered. Remissions are durable. We see activity in people resistant to treatments, not just had other treatments, but failed to dupe the anti-JAK inhibitor in one case, cytotoxic. So we may be in an era where maybe we don't need to treat you for a year, and unfortunately you stop your drug in a year and 2 weeks later the disease is back. Okay. If you look at all those studies that Albert alluded to, the rebound occurs in 4 weeks or less. That's why we didn't have to follow patients for more than that. Really wasn't worth it. We weren't seeing any rebound, and every other agent, the rebound occurs within a couple of weeks. And then finally, just again to remind you, we are enrolling a phase 2 randomized double-blind trial in atopic dermatitis. So really quickly on that trial, I've shown this before, 12 weeks of therapy, moderate to severe AD, very similar to the patients that we just described to you. 4 arms, placebo, 200 milligrams QD, 200 milligrams BID, 400 milligrams QD. So we are looking at QD dosing, and of course the placebo. They're equally balanced. It's completely randomized. It's a global study. The primary endpoints will be the typical what's your EASI score at 12 weeks, and then of course the other secondary endpoints. And this study is now ongoing. We expect to have data from this study around third quarter of 2027. Now, we have a very important collaboration with a company called Angel Pharmaceuticals. I'm pleased to announce that Dr. Charles Lee from Angel, he's the president of Angel, he's my boss, is here with us today. Charles and I and the team at Corvus and the team at Angel work very closely coordinating research and development. Angel has the rights to Soquelitinib in China. We have the rights in the rest of the world. And this is the study Angel is doing. It's pretty similar to what we've been doing. 12 weeks of therapy, moderate to severe AD. There are 2 cohorts. It is placebo-controlled. We think placebos are important in this disease, even for the laboratory measurements. And basically 100 milligrams BID, 200 milligrams once a day, placebo. We can look at the data then they go into the next cohort, 200 BID, 400 once a day placebo. We will have data from the first cohort, Charles tells me, or I guess he's going to be in trouble, by the end of the year. The Angel team is doing a spectacular job. Again, we work very closely with them. We have conference calls every week going over patient selection and information and so forth. Once that's completed, the study automatically rolls over to phase 2, 60 to 90 patients treated, again, with a placebo, treated on one or 2 of the doses that are studied. So again, sort of to summarize, we think our results show that Soquelitinib could become, I say leading oral, I mean leading therapy. It is oral, but I think a leading therapy period for atopic dermatitis and possibly other diseases. And I won't go through all these diseases. We have positive clinical results. We have shown safety. We see deep responses, including in patients who are refractory to other therapies. That is very challenging patients. We see durability consistently. Now, are we going to see 100% durability forever? Probably not. I'm sure that there's going to be patients who fail at some point in the future, but we haven't seen it yet. And we have, I think, identified immunologic parameters that support the mechanism of action that are going to teach us more, that are already informing our second and third generation compounds. Okay, this is an iterative process. Take the patient, go back in the lab, make better stuff. And I think there is a possibility of coming up with biomarkers that maybe will predict who does well, who has the more durable responses. Maybe it's a Treg, maybe it's a cytokine from the Treg or something like that. So there's a lot of work to do, but we have something very special, and we're all over it. So finally, I use this bullseye slide to remind myself, I look at this every night, that it's the target. It's all about picking the good target. A target that has applications to a disease that's relevant and hopefully more than one disease. And we've been able to make a very specific drug, the aha moment, the inventive step, go for specificity over potency. We used to argue, hey, this one's more potent, but I would say, yeah, but it hits RLK. No, I don't care about the potency as much. I like that, but I want the selectivity. I want both. Get that. And as you can see here, phase 3 in lymphoma, interim data from the lymphoma study later this year. I forgot to mention Angel data. Maybe I did mention late this year. So we're really looking forward to the Angel data in their phase 2 later this year. Angel's also going to be starting an asthma study, and I know they're preparing to file an IND on that, as we are. And then I think you can see the rest of the opportunities here. ALPS, I didn't have time to talk about that. We are planning to hopefully present some of that data at the ASH meeting in December. That's a very interesting disease. Very rare disease, but very interesting. Again, teaching us a lot. Autoimmune lymphoproliferative syndrome, bad disease. Finally, our upcoming milestones: phase 2 HS study, asthma by end of year, Angel data late 2026, ALPS data, ASH, phase 3 interim data later this year, and lymphoma. Those are the near-term milestones for Corvus. And so with that, let's open it up. Maybe Dr. Sarin and Dr. Chu can come to the front and we'll answer questions. I think we'll take the questions from people in the room. And then we can also open it up to the webcast. So any questions for any of us? Mostly ask Albert and Kavita because they're smarter. Greg.
Yeah, Greg Sloan. Oh, sorry. It's Greg Sloan at Mizuho. Thanks for the presentation. Thanks, doctors. I think investors who look at this data see it as early. It's promising, but it's early. We've got other programs that are out there as well. So, and there were some references to JAK inhibitors and STAT6 and perhaps some other mechanisms of action. But as maybe for Dr. Chu, as you are involved in many clinical trials right now, as you see the ITK inhibition mechanism of action relative to the other mechanisms of action, is there any other aspect of what you're seeing with Soquelitinib that beyond maybe the prevention of rebound or having long durative efficacy, at least at this stage, is there anything else that you would point to that helps to differentiate this versus some of the others where investors seem to be paying attention to equally, if not more? So.
Yeah, thanks for the great question. Honestly, I was holding bated breath for the safety data to come through. I think, just like I started with, I think safety is paramount in this disease. I think a lot about these pediatric patients that will eventually need something. And then on that particular point, it's great that we have dupilumab, which is a great treatment, but it's an injectable, which for kids is a big deal. So a safe oral medication, I think just that alone is something right there. I think the efficacy data, yes, really, really early. But just so promising, right? Especially looking at that 2-month dosing, the 75% patients hitting EASI 75. I mean, you can put that up against most of, these really promising investigational agents that are coming through right now. So I think in terms of oral medication, where you have just checking that safety box, at least from what we know, and again, really hoping that holds up in later phase trials, with that really promise of the efficacy. That's fantastic. Honestly, durability so fascinating. That was icing on the cake, right? To come in after that. But I was talking to someone earlier. If you kind of look at that cohort 4, 2 months of treatment, and then maybe 90 days of durability. I apologize. That's cohort 3. That's like about 6 months, right? You can imagine, even if that's the worst case, and it just that durability plummets at the end of that. Like 2 courses a year and that sort of response for AD, boy, that's a great conversation to have with the patient.
Thank you. Next question.
Hi, Tony Rowe. First of all, I think the durability issue is, getting back to the last question, nobody wants to take this medicine continuously. And so having the durability is the thing that really distinguishes this. It's amazing. So, but I noticed that the highest dose also was the only dose that had blood levels of Tregs. And also, it seems like the more you go out, the longer the durability. So, what are your plans for, is it possible then to take it 2 months for a year? And what do you think about that? I mean, because that is a game changer.
Thank you. First of all, we should not let that person in the room. He's a professional. But can I take that? So, Tony, Dr. Oro, this is the data we have now. So, 1 month gives us Tregs that persist at this dose. 2 months gives us Tregs that persist. I think a real question now is longer treatment give you even greater durability? Maybe you don't even need to give it for 28 days. In animal models, you don't need to give it that long, and you may induce this. So that's another thing to think about. Can we even go shorter and generate the Tregs? So we don't really know the answer to that. But in animal models where we can be a little more we can do more biopsies and look around. There's no question. You see Tregs infiltrate lesions, skin lesions, very quickly. And same thing in asthma models. That's new biology, and I think there's going to be a lot of clinical questions. But I think from a clinical development standpoint, not to worry. I think the clinical paradigm for approval would be 4, 8, or 12 weeks of therapy, placebo-controlled. What's your EASI score? Now, if you want to claim I can retreat somebody a second and third time, I think you are going to have to go in the clinic and prove that. But that's been done before, with Dupy and so forth, continuation studies.
But the question was, why is it different from everything else? And that's the answer, is that it's the durability, non-injectable durability is a game changer.
Yeah, thank you for that comment.
That's right.
Any other questions? Zach?
Yeah, we have a couple from the webcast here.
Okay.
So I'll start with one from Paul Choi from Goldman Sachs. For the doctors, what sort of duration or study size is needed to establish the benefit-risk profile as safe?
Yeah, so it's a great question. So I think one of the things that maybe we'll have to talk about is the duration of the clinical trial, right? So this phase 1, for a lot of the patients, only 30 days of follow-up after that dosing period. I think what we're seeing is that there is a pharmacodynamic effect that's persisting with that durability, which is a great finding in this trial. But I think we do have to follow these patients out many months. And I'm starting to think about the other trials that have been run for, say, the biologic trials or others to just get a sense of that safety over that full exposure period of months after the last dose, which is a great opportunity as well to further evaluate that durability aspect of the medication as well. I know that as we've been talking about the next phase trials, a lot of that is powered around efficacy endpoints as well. So I think maybe if you want to touch upon some of those considerations, but at least what I'm thinking about is this drug is really interesting even after you stop the last dose, and that having some of that safety follow-up for, throwing out a number of, 6 months afterwards, et cetera, just really understand what's going on there. Because again, we haven't seen the end of the durability yet, at least from these initial phase trials.
Maybe if Paul has a follow-up question on that. So, because I wasn't sure whether he was asking about the safety in the off-treatment period or the on-treatment period. I mean, both are important, but I forgot to mention that in the follow-up period, the study is still blinded. So the patient and doctor still don't know what they're getting during the follow-up. That's an important point. One other point, as long as we're talking about placebos and controls, is as you can see from Dr. Chu's safety slides, there's really no side effects that are, that are worrisome to the patient. The placebo and the treatment were identical, basically. And this is an oral medication. So when we say the trial is blinded, it really is blinded. So just think about if you were taking a drug that had injection site pain, and let's say it was 60 or 70% of patients. Well, that's not really a blinded trial because the patient knows that they're getting the drug. And I see this, to me, this disconnect when in some of these trials where, you got 20% of people getting conjunctivitis, you've got 60% injection site abnormalities, and they occur every time. And so how do you blind that? Well, the answer is it's not really blinded. Now, I'm not criticizing it. It's the best you can do. I mean, I think those studies are doing the very best they can do. But it really does raise the question. But when we say blinded, I can tell you, because I've swallowed some of those pills, they're not different. Okay? I don't want to do it for a year, but they're not different. And it's impossible to know what you're getting. So it really is blinded. Anyway, any other questions from that?
Yeah, we can move on to another one from Lee Watzik from Cantor. This is a three-part, so bear with me. Immune markers, cytokines were lowered in the drug-free stage. What would be the explanation for that? Do you see that reduction from the placebo arm? Next, can you discuss what you saw in placebo arm in terms of rebound and biomarker changes? And then last, how does the biomarker data read through to other potential indications for soquelitinib, the asthma trial, HS, etc.? Can we expect to see similar durability effects?
Okay, so I hope I can remember the three questions. The first question is the reason the cytokines are still falling in the drug-free period is because there's Tregs around and the immune response is still being, inhibited. So that's our explanation for why they continue down. Now, you'll notice they bounce around a lot, cytokines. Measuring cytokines in these patients is very difficult because the timing is difficult. I mean, every patient's a little different. You don't know when the optimum changes are occurring. Some of these cytokines are very short-lived, like I think IL-4 has a plasma half-life of 4 hours, so you can easily miss these things. We are noticing interesting things like IL-5 drops early, IL-4 later. Now, what is the explanation for that? We're not sure. Different subsets, as Kavita mentioned, different subsets of cells being affected differently. So the answer to the question number one, the cytokines continue to drop because of the ongoing Treg-induced immune regulation. The second question on placebos. Placebo cytokines jump all over the place and really don't give you any constant stuff. They could be down, they can be up, they can be down, they can be up. It's just really, it didn't show that really for the sake of simplicity. You certainly don't get a consistent thing in a drug-free period. There's no question about that. Now, the rebound question on placebo, if you remember those slides that Albert showed, placebos don't rebound. I always get asked that question. How come placebos don't rebound? Well, because they don't respond that well. Remember, we had 0% EASI-75s, and they didn't get any drugs. So how, in cohorts 1 and 2. So if you don't have much of a response, it's hard to rebound. And they're not getting treated with anything. So whatever the mechanism of rebound is in a JAK inhibitor or DUPI or a STAT6 inhibitor, whatever that mechanism is, I wish I knew what the mechanism is, but I don't think anybody knows. But it's really striking how quickly it occurs. Within a couple of weeks, if you look at those curves. So anyway, the question of do placebos rebound is sort of not really addressable because they don't rebound. There's nothing to rebound to. And then I think the other question was
the read-through for other indications.
Oh, well, we think the read-through is good because, and that's why we're doing an asthma study, which again is thought to be Th2 driven, and all the cytokines and things that we're thinking of are important in asthma. Now, we are going to do something a little different in asthma, and that is, given our mechanism of action, we do not need to restrict it to eosinophilic asthma. So let me say that again. That's double the population, if you don't, roughly. Because half the patients don't have eosinophilia. So most asthma studies require an eosinophil count of 300 or sometimes 150. That is eosinophilic or allergic asthma. We're going to open that up wider. And so I think the read-through is, all I can say is that we've now looked at many different animal models: inflammatory bowel disease, asthma, psoriasis, atopic dermatitis, graft-versus-host disease, systemic sclerosis. I may be forgetting a few, and we see consistent efficacy, robust effects in all of those diseases.
Okay, next one from Etzer Darout from Barclays. Have you also looked at the placebo group EASI scores in cohorts 1 to 3 out to day 118? And also, do you have 90-day off treatment for cohort 4?
So let me take the last one. We don't have 90-day treatment for cohort 4 because we recognize that we didn't really need to do it. First of all, you got to realize to do this requires an amendment to your protocol, IRBs, and you also have to pay for this. And we were comfortable enough with what we saw with the Treg data from cohort 3 and from cohort 4 to say, okay, let's take our resources and double down on this in the phase 2 trial, which we're doing, and where we will have 90-day follow-up. Okay. Now placebos flare. If you follow them in these drugs, I don't think we went over the flares, but I don't think we've ever seen a flare in a treated patient, but we do see it in the placebos. So placebos bounce around, as you know. Also remember another thing about our trial. Somebody else, I think, Greg, you asked this question. I would argue that the patient population we have is sicker than most of your AD studies. Why do I say that? First of all, 35% of prior therapy. 8 patients have failed or refractory to prior therapies. Nobody takes those. Nobody takes those patients. So when you talk about a third of the patients and half of those are refractory in a study with only 50 subjects, that's a pretty big hit potentially. And so we don't have 1,000 patients, and you need to do that at some point. But we have a very robust clinical experiment where we took patients who were pretty sick, and we saw efficacy. That's what motivates us to go on. Look, I think that the data we've shown in a sicker patient population is as good as any data, any product under development I've seen. And even some of the ones approved, albeit they have, hundreds or thousands of patients. So that's why we're excited about this. That's what we're doing.
Okay. Next question from Sean Lee from H.C. Wainwright. Seeing the inhibition effect on JAK-STAT pathway, would that potentially lead to safety issues long term? And does it matter to tease out the treatment effect from the Treg effect, or does it not matter?
Okay. So I'm glad that he asked that question, because I forgot to mention that remember, it's not like we're blocking JAK-STAT in every hematopoietic cell. Okay. It's only in the cells that have ITK and probably only in those activated T cells. So it's very restricted. Now, again, the fact that we're seeing changes in the JAK-STAT pathway, it's not because our drug is interacting with ITK. Let's make sure people understand that our drug does not bind any JAK 1, 2, 3. No. So this is an indirect effect. And it's due to the fact that, and it's very clear in the patients that we treat with soquelitinib, SOX3 stands for suppressor of cytokine signaling 3. It is one of the brakes. Remember, the immune system has gas pedal and a brake. So SOX3, when you're trying to shut down an immune response, inhibits the JAK-STAT signaling because you're trying to cool things down. So we're seeing patients' inflammation go away. We're blocking T cell signaling upstream. Naturally, SOX3 goes up. SOX3 is going to block those pathways. So it's almost a secondary effect. I don't think it's a primary effect. But again, it's nice to see this biology happen. Right? It's nice to see that you're having this multitude of effects in conjunction with clinical responses and durability and all that other stuff. But just to be clear, we're not a JAK inhibitor. Because we got, some guys out there saying, oh, we're a JAK inhibitor. No. We're not a JAK inhibitor. We don't bind JAK. No binding to JAK. We affect SOX3. SOX3 will dampen some of the JAK-STAT signaling. Okay. And that's occurring in only the cells that have the ITK target. So that's not happening in your myeloid cell or your macrophage or any other cell. Okay, so that's very important. But it's very interesting to see that you really are shutting down this inflammatory response. You got Tregs. Well, first of all, what do you think Tregs are doing? They're trying to shut down the immune response. Wouldn't it be crazy if the immune system makes Tregs and turns on JAK-STAT signaling? You wouldn't, when you drive your car, step on the brake and step on the gas at the same time. Well, maybe you would, Jamie, but you wouldn't do that. So the immune system is dampening the response. Tregs and reducing JAK-STAT signaling. So that's an indirect effect, but very interesting because yet it's another clue to the fact that we have some very interesting biology going on. Unprecedented. Nobody's seen stuff like this before. Okay? Any other questions?
We'll do one more, and then I think we need to wrap up. Jeff Jones from Oppenheimer. Can you speak to what magnitude of change is typically considered clinically significant in regards to Th2 populations?
Th in general.
Well,
I mean, I think
that's a tough one. So I believe that Jeff is asking the relationship between the degree of Th2 reduction and clinical response. We just don't have enough data to make those kinds of correlations at this point. But that may be actually something, to look at. Listen, we're working really hard looking at all of this. Serum cytokine relationships to response, Treg in relation to response, all of those things. It's tough. You can't do, you're going to need many more patients to make those kinds of correlations. And what I said, this is human biology. I mean, it's highly variable. As you can see, these patients are, some are young, some are old, all different prior therapies, etc. But that's a great question, Jeff, but I don't think we have enough data yet to come to that. But should you get that, should you come up with the Th2 level that predicts response or duration of response, I would file a patent on that in about 10 milliseconds. Any other questions? So anyway, first of all, I want to thank Dr. Chu and Dr. Sarin on really wonderful presentations. I mean, that was really exciting. I'm so inspired now, I don't know what to do with myself. But I want to thank the people in the audience here for attending. I want to thank everyone on the webcast. I think that we have some really exciting data that we presented this morning, and Dr. Chu's presentation on Saturday, poster tomorrow, Kavita's poster also this afternoon. Again, you have a chance to come by and hassle her even more. So we look forward to seeing you at these posters, but I want to thank everyone, and we're eager to report on our future progress. Thank you.